So to start talking about the vaccine, we need to first review the COVID-19 viral life cycle. So the infection starts when the virus gets into your, for example, lungs. And so this white part here would be the space in your lungs. And then the gray part here is one of your lung cells. So COVID-19 has these spike proteins on the outside, and they bind to ACE2 receptors, which is a protein on the surface of different types of human cells, but lung cells have them. They usually do a different job in lung cells. I don't actually know what their normal job is, but they're there for a reason. They aren't just there to pick up viruses. So when the virus binds to this receptor, it causes the cell to move its membrane around the virus and take it inside the cell. So the virus comes into the cell and it releases its RNA into the cell, and the viral RNA gets reverse transcribed into DNA and then transcribed and translated into proteins. And the virus kind of takes over the cell, and the cell stops transcribing and translating its own DNA and tends to just transcribe and translate the virus's DNA. So the cell just turns into a virus factory. It starts assembling new viruses and then popping these viruses out, either a few at a time or just by the whole cell exploding with viruses. Kind of disgusting. This cell is going to start making more and more and more and more viruses until it dies. And these viruses then are getting released into the surrounding tissue and your bloodstream. So once these viruses are in your body, in the tissue replicating, there's a few things that happen. And this is kind of the same picture. Well, it's a different picture, but it's the exact same process that we talked about on Monday. So the macrophages, this is actually they are calling it a APC cell, antigen presenting cell. It's basically the same idea. So these macrophages gobble up viruses and then present parts of them and say, look what I found, look what I found. This is not me, this is not self, this is an invader. And so if they can't take care of all the viruses themselves, they call in the helper T cells. The helper T cells assess the situation and decide if they need to just call in more macrophages or if they need to call in B cells to make antibodies or killer T cells to destroy the infected lung cells. So this whole process is the same process that we talked about on Monday. Eventually you get long-lived memory B and T cells that will recognize the virus and patrol the body for months and years, providing immunity. Hopefully a long time, although we know with other coronaviruses, they only last a few years in humans. So these antibodies, what is neat about them is that some of these antibodies are considered neutralizing antibodies, which means they disrupt the initial interaction between the spike protein and the ACE2 receptor on cells. And so if you are making a type of antibody that is neutralizing, that really protects you because it doesn't let the virus get into your cells at all. But having any antibodies is still better than nothing, and it's going to attack the virus and alert the macrophages to the fact that the virus is in town. And so they can start getting the immune response up and running sooner than later. So we talked about the primary immune response versus the secondary immune response. And the second time you see a pathogen, the response, you get antibodies produced faster and you get way more of them. So that is the point of having memory cells, is that these memory cells can just quickly proliferate and say, aha, I am the memory cell that recognizes this virus, divide and divide and divide and divide, mitosis, 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 make lots of B cells that are antibody factories that can release antibodies into the blood that can disarm identify and potentially disarm the virus. So the whole point of making a vaccine is so that you don't have to get sick for this primary immune response. So your primary immune response happens the first time you are exposed to a pathogen. And so the idea is to have this happen in a safe way. And the safe way is a vaccine, because we know if we inject you with a vaccine, you're not going to get all the way sick, but we can trick your immune system into developing a response and developing antibodies that are specific to the virus. So the first question that I think people are asking is it going to be possible to make a COVID-19 vaccine? I think this is a good question, right? Where the entire world is banking on the idea that we're going to eventually be able to make a COVID-19 vaccine. And so first of all, is this a reasonable proposition? And fortunately, it seems likely that a vaccine should be possible to make. And the reasons that I think this is because, and scientists think this, is because vaccines for the original SARS and also MERS have been developed and tested in animals. They haven't really gone through the whole process of getting FDA clearance and trials and everything, because no one was really interested in SARS and MERS once the two epidemics passed. But when these vaccines were developed in animals, they usually resulted in protection from the virus and greater survival rates. Some of the vaccines, however, did result in complications such as liver damage or lung damage. So there is good reason to be somewhat skeptical about these vaccines and not just rush into making them at mass as soon as we get the first results in that they are working. We need to make sure that they're not going to result in complications in a lot of the people who take them, because a lot of people in the world are going to be taking this vaccine. When we look at infections from other human coronaviruses, there are 4 human coronaviruses that circulate every year as the common cold. 4 of them cause about 20% of the common cold. We humans, when we get infected with one of these viruses, it seems that we get protected from reinfection for a few years. And this jives with what we saw with SARS and MERS. Sorry, this should be SARS-CoV-1, not 2. Antibodies for these 2 diseases seem to last about 2 to 3 years in the people who are infected with them when those epidemics hit. Right now, one of the big issues that people are thinking about is, okay, once we have a vaccine, how are we going to make a few billion copies of it? And currently there's no large-scale manufacturing in place that can do this. There are companies that make flu vaccines, but we don't need nearly as many of those every year as we're going to need COVID vaccines. So people are working to start scaling up manufacturing and processes so that they will be able to make these vaccines once they exist, which is challenging right now because we don't know yet what is going to be in the successful vaccine. There's different types of vaccines which we're about to talk about, and making them involves much different processes for depending on what type of vaccine it is. So people are starting to think about how we're going to be able to do this manufacturing, but it's really hard to start moving on it too much because we really don't know what we're aiming for yet. We don't know which type of vaccine is going to be successful. So what types of vaccines are there? There are 4 major different types of vaccines. There are ones that contain the full virus. There are ones that contain a viral vector, which is kind of engineered spliced virus, a hybrid virus that has some parts from the virus that you want to make an immune response to, and some parts of a virus that we're just using as kind of the vehicle to carry the pieces of the virus that we care about. There's nucleic acid-based vaccines, which there aren't really, I don't think there are any nucleic acid-based vaccines in circulation right now, but people have been trying to develop them for quite some time now. And some are looking promising. And then there are also protein-based vaccines where the vaccine just contains one or more proteins from the virus, but not the full virus itself. So we are going to be talking about, in this mini lecture, the virus-based vaccines and the protein-based vaccines, which covers quite a few of the vaccines that are in development. So virus-based vaccines contain the full viral particle. So we can't just put viruses into people, right? Because that would just get them sick. So there's no point in vaccinating if you're just going to expose people to viruses. So we need to weaken or inactivate the viruses somehow. So there are two different types of full virus-based vaccines. There are weakened viruses and there are inactivated viruses. Both of them are full intact viral particles, but they've been weakened or inactivated somehow, and those two processes are pretty different. So I'm going to show you now. So a weakened virus is a virus that normally replicates inside human cells, like SARS-CoV-2. And then this weakened, or another word for weakened is attenuated. This attenuated virus is made in a lab so that it is not able to replicate in human cells, but it is able to replicate in some sort of non-human cells. So sometimes we use chick embryo cells, which are found in eggs. Influenza vaccine, there is an influenza vaccine that's very common that is made this way, which is why some people who are allergic to eggs can't get certain types of the flu vaccine. Sometimes people use insect cells. There's anyway some other sort of animal that the virus does replicate in. And so you can infect the animal with this attenuated virus. The virus will replicate and replicate and replicate in those cells, and then you collect out the virus. So this is live virus that can infect cells. It just can't infect human cells. So then when you give that virus to humans, the virus it looks a lot like the normal infectious human version of the virus, but it can't actually get into human cells and or replicate inside them. So an attenuated virus is a real live virus. It just is not able to replicate in human cells. So it cannot start an infection, but it looks just like a virus, so your immune system will mount a response against it. There are also inactivated viruses. So inactivated viruses are the real live human version of the virus, but it has been treated with heat or chemicals such that it is non-functional. So there are formaldehyde is a common chemical to treat viruses with, and that basically forms a bunch of extra bonds around the virus so that the virus can't come apart when it gets into cells. So these inactivated viruses can never reproduce anywhere. But making them is somewhat dangerous because you start with real live active virus that can infect humans. And so if you're making these in huge batches, you have basically vats of human coronavirus, which is kind of scary. So the manufacturing processes for these have to be super, super careful so that none gets out and infects the people who are doing the manufacturing. But once the virus has been inactivated, then it's no longer able to infect people. But again, it looks just like the regular infectious virus. So when it gets into your body through vaccination, your immune system will again mount a response and start making T cells and B cells and all that, and antibodies against it. Okay, the other type of vaccine I want to talk about is the protein-based vaccine. And so in a protein-based vaccine, proteins from the surface of the virus, so for COVID-19, that would be the spike protein. These proteins are made in isolation without making the whole virus. So usually this would happen by making the proteins either in bacteria or again in some sort of cell line like insect cells. And this happens the same way that what we were talking about for the spider goats or papaya ring spot viruses. So you take the gene for the protein and you splice it into the bacteria's genome or the insect cell or whatever type of cell you are using for your production. You splice the gene for the spike protein into that cell that you're doing the manufacturing in. That cell will then start making the spike protein, then you'll purify out the spike protein, and then that's your vaccine. So I want to tell you a little bit about where we are currently on vaccine development. So as of May 22nd, there was a report by the WHO, and they are tracking all of the vaccine candidates. So of these candidates, 114 of them are currently under exploration or preclinical trials. This means that they're probably being tested in animals. That's what preclinical trials are, or they're being explored, which means they're still being designed and tested in a lab in assays in cell lines, not in animals. And there are 10 that are currently in phase 1 or phase 2 clinical trials. So when I'm saying phase 1 and phase 2, what does that mean? So there are phases of clinical trials, whether you're developing vaccines or drugs or diagnostics. The FDA has different phases for their clinical trials. So most vaccines for COVID right now are in phase 1 or maybe phase 2. Most of these 10, which is not very many. So in phase 1, you recruit somewhere between 20 and 80 healthy adults. These adults are healthy. They're like ideal candidates for a vaccine to be successful in, and ideal candidates who will not get complications or get very sick if something does not go well. So the point of phase 1 is to assess the safety and the immune response, and determine dosing and how the vaccine will be administered. So these healthy adults are going to be tested, watched very closely to make sure that they're not getting serious side effects. And then their immune response will be measured. So their blood will be taken, and then their antibodies will be taken out of their blood and tested to see if they work to neutralize the virus. In phase 2, it looks pretty much the same, but it's a lot more people. And then this is a controlled study with a placebo group. In phase 1, everyone who gets the vaccine is getting the real vaccine. In phase 2, you have a controlled experiment with a placebo group, which means that half the people are not getting a real vaccine. They're getting saline solution or something. So again, we're assessing safety and the immune response. So they're testing for if you're making antibodies and if there are bad side effects. And again, optimizing the dosing and the vaccine schedule. They're figuring out if you need to get boosters. Then phase 3 is a lot more people. So we are going up to 1,000 to 10,000 people, and this is a heterogeneous population. So in phase 3, you want to start pulling, we're pretty sure the vaccine is safe, so you want to start pulling in a lot more people that are more representative of the population that you're going to be giving this vaccine to. So interestingly, even vaccines that are meant for children are still, phase 1 and phase 2 are still in healthy adults. Or vaccines that are meant for elderly people are still going to be given to kind of middle age healthy adults in phase 1 and phase 2, but phase 3 is when we start actually looking at the full population that we're going to be giving the vaccine to at some point. This is a double-blind test against the placebo, which means that the doctors who are administering the vaccine and the people who get the vaccine are not going to know whether or not they get the real version or the placebo. Here we'll assess effectiveness and actually see if it prevents the disease. So we'll look at these people and see if they're out in the population, out doing their thing. If they are exposed to the disease, are they protected from it? And so basically we do this by looking at how likely is it for our vaccine group versus the placebo group. Is it more likely that the placebo group gets sick, or is it more likely, or is it equally likely that the placebo group and the vaccinated group get sick? So the point of the placebo is to see that whether the vaccine is more protective than doing nothing. The placebo basically means that you're recruiting people that are not getting vaccinated, so we have something to test against. The placebo is our negative control. So we want to make sure that people who are getting the vaccine are protected more than the placebo group. But we also want to make sure that any side effects that we're seeing from the vaccine are happening at an equal rate in the placebo group. So if people, for example, are getting lots of headaches after they get a vaccine, is that because people just get headaches, or is that because it's coming from the vaccine? And the only way you can know that is if you have a placebo group of people that you're testing for exactly the same things, but have not gotten the vaccine. So we're assessing effectiveness and disease prevention, and they'll be observed for longer periods of time so that we can find side effects that are more rare. Once you've done all of this, then you can submit your data for FDA approval, and then you can start going out into giving your vaccine to the full population. So the big crux here is that you need to observe people for a long period of time to make sure that there aren't side effects that pop up later, or that there are side effects that are only observed in a very small fraction of the people who get the vaccine. And so this is the concern with pushing through these vaccine trials faster than they should be. So already these trials are going at lightning fast speed. There's like the FDA approval process usually takes, it's like mountains of paperwork and usually takes forever. And the FDA is flying these things through the process. But there are some parts of the process that just take a while. And that is, once you give someone a vaccine, it takes a while for your immune system to mount a response. It takes a while for us to be able to see whether there are side effects. And these are the things that you really just can't speed up, no matter how much somebody wants vaccine trials to go faster. You just need to wait these periods of time to test, to wait and see if the vaccine is effective or if there are side effects, and make sure that it is safe. The worst thing we could do is get a vaccine that ends up having a huge amount of side effects in a big portion of the population, and then having people refusing to get the vaccine. And then imagine we do get a vaccine that doesn't have side effects at some point later down the road, nobody's going to trust it. So it's really, really, really important that we are not worried, that we are very careful about this whole process and we aren't rushing through it. We need to make sure that the science is being done properly and following all of the guidelines that the FDA has in place to protect people. So this is the list of all 10 of the vaccines that are currently in trials. Some of them are in trials in the US, some of them are in trials in China or in Europe. And so I'm going to tell you a little bit more about 2 of these vaccines. One of them is an inactivated vaccine, like we talked about. There's only one protein subunit vaccine that's currently in trials. And then there are also some RNA and DNA RNA and DNA vaccines that we didn't talk about how they work, but I want to talk about one of them because this is the only one that has any data out yet from human trials. Okay, so the first vaccine I want to tell you a little bit about is an inactivated virus vaccine from a company in China. And this company recently released some results that shows in preclinical trials, so they gave their vaccine to monkeys to see what their immune response was. It does protect monkeys from getting infected with the virus, which is great news. So this is a quote from the article. Researchers from Sinovac Biotech, a privately held Beijing-based company, gave 2 different doses of their COVID-19 vaccine to a total of 8 rhesus macaques. 3 weeks later, the group introduced SARS-CoV-2, the virus that causes COVID-19, into the monkeys' lungs through tubes down their tracheas, and none of them developed a full-blown infection. So this is really great news. Of course, it's only in 8 monkeys, so this is still very preliminary. We don't know how well that's going to translate over into humans, but it's some exciting news. There's And then this is a little bit about Moderna's vaccine. So this is an mRNA-based vaccine that's in the US. And they have now looked at the results of 8 participants. All 8 of them began producing antibodies, which is excellent news. And they tested 4 of these participants to see if they developed neutralizing antibodies. And so far they've only tested 4 of the 8. They're working on testing the next 4. And the 4 that they have tested did have neutralizing antibodies in their blood that were at levels seen in recovered coronavirus patients. So again, this is really exciting, but also still very preliminary. There is, like Dr. Shockett was saying, there's a lot of great information coming out on Twitter about these things, this data. But an interesting person to follow is this scientist named Ian Hayden, who studies proteins, so we know he's cool. But he was one of the 8 participants who got this vaccine from Moderna. And so he's been talking about his experience and also staying on top of the data coming out from Moderna about their vaccine. So I'd recommend checking him out if you're interested to see what it's like to be in a vaccine trial. Okay, so let's talk about these different vaccines. So someone had a question earlier about, is it possible to reactivate the virus, an attenuated virus in the body? So that's part of this question, so we can talk about that now. And we are, excellent, we are all over the place. So let's talk about this. So first of all, the definition of an attenuated virus is that the virus can only reproduce in non-human cells. That's because the virus has been evolved so that it will no longer reproduce in humans. So it's definitely, we can knock out B, and we know C is definitely true. That's the definition of an attenuated virus. So here's a question though. Is there any chance that the virus could replicate and develop mutations that could make it infectious to humans again? And it is technically possible for this to happen, but it is extraordinarily unlikely. And so we talked about this when we were talking about the chances of developing mutations are, first of all, very low, but also developing the exact right mutations all at once to make this virus infectious. It's kind of like developing all of the mutations that you would need to get cancer, right? It has to happen in a sequential process. And you have to get exactly the right ones. And so it is extraordinarily rare. It is extremely unlikely that A could possibly happen. But in theory, it is technically possible. So I'm not going to ask this on a test of you, but I just wanted to talk about A and point out that in theory, it is possible that an attenuated virus could develop mutations because it is a real live replicating virus. However, it is extraordinarily unlikely that it would end up being infectious again because of the number of mutations it would need to develop. Okay. Which type of vaccine contains whole viruses? All right, we've got most of the class on D, some on E. So yes, attenuated and inactivated vaccines are both full live viruses that have either been, for attenuated, made so that they cannot reproduce in human cells anymore, or B, have been chemically treated so that they are no longer active viruses. They can't reproduce anywhere anymore, but they are full viruses. So A and B both contain full viruses. Protein-based vaccines only contain a protein or maybe a couple proteins from the virus, but the virus is not intact. It doesn't have any of the nucleic acid. It doesn't have any of the membranes. It is just a couple of proteins. So C is definitely not a whole virus. It's just a few proteins. So I would agree with D in this case. All right, let's skip this. So I want to talk about, finally, about COVID-19 is antibodies. And I think this will potentially get to this question. So antibodies, again, are specific to a little piece of a pathogen. So they have these variable regions at the tips of their Y-shaped proteins, and in that little tip binds an antigen. And an antigen is a little piece of a pathogen. So these variable regions bind specifically to exactly one antigen. Both sides of the antibody bind to the exact same antigen. And then there's a constant region of the antibody that can interact with other cells in the immune system, like macrophages. So macrophages interact with this part of the antibody, and when they do, they suck up the antibody and whatever the antibody is bound to, which would be the pathogen, and then they gobble them up. So antibodies can be collected from people who have already been infected with COVID. So if someone had COVID and then recovered, they presumably have antibodies that protect them from getting COVID in the future. And so doctors can draw their blood and then purify out just the antibodies to create what's called a convalescent serum. And then this serum can be injected into other people, either as prophylaxis, which means preventative treatment, or as therapy for people who are already sick with COVID and aren't yet making their own antibodies. So if they're early in the process of the infection. So this is super powerful as a way to prevent and treat COVID. Obviously, it's not really possible to do this at scale because it requires taking blood from people who are recovered from COVID. Like it takes a fair amount of blood to make a treatment for one other person. So this isn't something that we can do at scale, but it's nice to have it as an option for the people who are super sick, or potentially for people who are on the front lines, like nurses and doctors. So I've been kind of dancing around this neutralizing antibodies thing, so I want to just talk about that for a second. So antibodies bind somewhere on to one of the proteins on the surface of the virus. And it's important where exactly on the protein it binds. And so if we look at this virus, so this is a picture of SARS-CoV-2 interacting with the ACE inhibitor, and you can see that only a part of the spike protein physically interacts with the ACE2 receptor. And the antibody is going to be specific to a place on the virus. So if you have an antibody that binds over on the side of the spike protein, this is going to help the immune system identify the virus and tag it for destruction by macrophages, but it's not actually going to prevent the virus from binding with the ACE2 receptor. However, if the antibody happens to bind on the surface of the virus, so these would be two different antibodies made by two different types of B cells. If one of them happens to bind on the part of the protein that should be interacting with the ACE receptor, then it's going to prevent the virus from being able to interact with the receptor so that it can't get into human cells. So this is a neutralizing antibody. It's even better than just these identification antibodies. These are the ones that you want to be able to make, that we need vaccines to elicit in us to make them preventative. So this is what people who are doing vaccine trials are looking for. They're looking for neutralizing antibodies that are going to prevent the virus from being able to get into our cells in the first place, not just being able to bind somewhere on the virus to flag it for destruction. We want this physical inhibition. So I think there's been a lot in the news about COVID antibody testing. And I think the media has actually done a really terrible job of blowing it out of proportion. So right now, COVID antibody tests can tell you whether you have antibodies in your blood that bind to the virus. If you have antibodies in your blood, that means you have memory B cells against SARS-CoV-2, which means you have been infected or vaccinated in the past. There are some antibody tests that can tell you whether you have neutralizing antibodies. These are the great ones that bind to the right place on the spike protein that stop the virus from being able to get into your cells and start an infection in the first place. So if you have neutralizing antibodies, this means that you're protected against future exposure. Only some of the tests are currently good enough to discriminate between these two different types of antibodies. Furthermore, many antibody tests have high false positive rates just because antibodies are tricky to work with. And so especially with the lower FDA regulations, FDAs have lowered their barrier of entry so that they can start expanding this testing quickly and not bog everyone down in paperwork, which is great, but it means that a lot of the tests on the market right now really shouldn't be on the market the way that people are using them. Someday, these tests are going to be good enough that they will be able to give us accurate information on protection, right? To see if are we immune to further infection. But right now, they're not good enough yet. So what we want to know is if we have neutralizing antibodies, which is a hard test to make, and it takes a lot of testing and going through the process to know whether we have these yet. So if you have the opportunity to take an antibody test, I would recommend it. But the only thing that you can infer from the answer is whether or not you've been infected in the past. You can't yet infer whether or not you have neutralizing antibodies. Okay, so there's a short answer question on Camino that basically just makes sure that you got that point. So that's, again, optional, and I don't have time for you to do it right now because we're at the end of class, but I just wanted to point out that there are clinical trials recruiting healthy volunteers, or also trials who are recruiting people who've had COVID-19 so that they can use their antibodies for testing and potentially convalescent serum. So if you're interested in getting involved in doing something, this is a great way to get involved. Some of them you get paid for. Also, Santa Clara County is recruiting contact tracers. Some of them are volunteer, some of them are paid positions, but if your summer plans fell through and you're looking for something to do, you might consider becoming a contact tracer. And there's information here on how to do that.